So this lesson is about the Krebs cycle, also called the citric acid cycle. It is the third stage of cellular respiration, and it takes place in the mitochondrial matrix where there is oxygen present. So we start with the end product of pyruvate oxidation, which is a 2-carbon molecule called acetyl-CoA. What it does here is it joins with a 4-carbon molecule called oxaloacetate with the help of the enzyme citric synthase to form a 6-carbon molecule called citric acid, hence the name citric acid cycle, after the first molecule formed. And then what happens to citric acid is it goes through an isomerization reaction with the help of aconitase to form the isomer isocitric acid, also 6-carbon. What happens here is that water leaves, H2O, leaves, and then actually comes back in. I'll show you here in the next slide where there's structures. We have citric acid, which had an OH here, and an extra hydrogen here. And then we ended up with isocitric acid, which is now, here that OH is no longer on the 3rd carbon. After water came back in, it got replaced at, the OH got replaced at the 4th carbon. And also you'll see the H is here, the other H, the other hydrogen. Hydroxyl group is now on the 4th carbon. The OH is the hydroxyl group, and the other hydrogen is on the 3rd carbon. So now what happens to isocitric acid? These are very important steps 3 and 4. They're very similar. You lose carbon dioxide molecule, so isocitric acid gets oxidized. And when something gets oxidized, you know that something else is getting reduced. And that's NAD plus is getting reduced to NADH with the help of a dehydrogenase. Remember, every time there's a transfer of hydrogen, a dehydrogenase is involved. Remember, a dehydrogenase transfers hydrogens. Same thing again. So when you lost the carbon dioxide, you lost a carbon and 2 oxygens, making it into a 5-carbon molecule. And again, same thing happens here. So you get a 4-carbon molecule, lost another carbon and 2 oxygens. And then here, succinyl-CoA gets converted into succinate. And in this process, you get GTP, which then gets converted into ATP. Now the difference between GTP and ATP is the nitrogenous base. Guanine triphosphate gets converted into adenine triphosphate. Well, what you need to know is that you produce ATP in step 5. And here's still a 4-carbon molecule, which gets oxidized. Okay, so it gives up 2 hydrogens to FAD, which gets reduced into FADH2 with the help of a dehydrogenase again. And that converts succinate to fumarate, also still 4-carbon molecule. Okay, and then water enters, in step 7, water enters and converts fumarate into malate. Now here, this is not an isomerization reaction, because here now malate has more molecules. Different than step 2, where water left and came back in, you still had the same amount of atoms. But here malate is a larger molecule than fumarate. And what happens to malate here now is that it gets oxidized, means something else gets reduced, NAD+, and you get NADH. NAD+ gets reduced into NADH. So then what happens is you get oxaloacetate again, to start the cycle all over again. Okay, because now remember, from one glucose molecule you will get two acetyl-CoAs, so the cycle will repeat again. For one glucose molecule it will happen twice. So the result of one turn is 3 NADH, 1 FADH2, 1 ATP, 2 CO2. But from one glucose molecule, everything's doubled. If I ask a question, what is the result from one glucose molecule? You will have to say 6 NADH, 2 FADH2, 2 ATP, 4 carbon dioxide. So the purpose of all this is to produce the energy carriers, NADH and FADH. Here you have the 3 NADHs, and this cycle runs twice. Here we'll see 6 NADHs, and the purpose is to produce these energy carrier molecules to send them to the electron transport chain to power up the electron transport chain to produce ATP, to power up the ATP synthase. The electron transport chain oxidative phosphorylation will be in the next video. So you will see here that you did get 2 ATP from glycolysis, 2 NADH from glycolysis, 2 NADH from pyruvate oxidation, and 6 from the Krebs cycle, and 2 FADH2 from the Krebs cycle as well. Okay, and notice that we get ATP from everything except pyruvate oxidation. No ATP from pyruvate oxidation, which is the second stage. So the Krebs cycle gives us the most NADH, because that's the purpose of it, to give us the NADH and FADH2 as well. And then the purpose of the electron transport chain is to give us the most ATP with the help of the energy carriers, NADH and FADH2. So here at the end I have a worksheet for practice, and I have the answers in the next slide. So coenzyme A detaches from acetyl-CoA and the 2 carbon molecules and the 2 carbon acetyl group to an existing 4 carbon molecule, resulting in the production of citrate. Citrate is converted to its isomer by the removal and then addition of, take a minute, pause the video if you need to, a water molecule. You'll see it here. The 6 carbon chain is partially broken down, releasing, take a second, pause the video, carbon dioxide. Electrons are removed, reducing NAD plus to NADH. More blank is released and more blank is removed via the electron carriers, very similar to step 3. More carbon dioxide is released and more electrons are removed via the electron carriers. One blank molecule is formed. Only 1 ATP is formed in the Krebs cycle. 2 hydrogens are transferred to step 6. Step 6 we have FAD, which is transferred into 1 molecule of FADH2. As you see. The addition of 1 blank molecule rearranges bonds in the substrate. Addition of water molecule. Step 8. The substrate is oxidized, reducing blank to blank and regenerating a 4-carbon molecule that starts the process all over again. NAD plus to NADH. And that's it for this lesson.